Everyone to the Ocean Insights seminar series. So today we have a presentation by Femi Delhamut, focusing on the South-East Madagascar Bloom, an observational and modeling perspective. So Femi completed his PhD at the University of Cape Town in South Africa in physical oceanography. He has then had two postdoctoral positions at GMR in Kiel, Germany, and also at the University of Dalhousie in Halifax, California. And Femi is currently a postdoctoral fellow back at GMR in Germany. And Femi is also the chair of the IIOE2 Early Career Scientist Network, which is the organization which hosting the seminar series. So it's a real pleasure to have our current chair presenting. So Femi, the floor is yours.
Great, thanks, Matthew. I hope everyone can hear me well. Is that, yeah, all good?
Yeah, all good from the side.
Okay, perfect. Yeah, hi everyone. I'm happy to be able to share some of my PhD work, which I did a few years ago, but which is still part of my interest currently. And so this work is on the South-East Madagascar Bloom, and it has two components: observational based, and then a modeling based component. So I will guide you through a brief introduction, not brief, a quite thorough introduction of this Madagascar Bloom, and then I will dive into the results. So I would like to start with this figure here, which shows satellite chlorophyll A concentration averaged during February 2006, and this figure actually shows the quite an exceptional view of this South-East Madagascar Bloom and its spatial distribution. So during its peak, it can cover a length of around 2,000 kilometers and a width of 1,100 kilometers, which is quite a large area for an open ocean bloom. So, of course, when phytoplankton blooms are quite important because they play an important role in biogeochemical cycling of elements. Of course, also the regulation of atmospheric CO2 and the marine biomes into this region. And this bloom here actually occurs in an oligotrophic region, and it, as you can see, has a very filamentous structure, which could indicate a possible importance of mesoscale eddies in driving this spatial dispersion of the bloom. So yeah, I said all of this already. So I would like to point out that so in December 2019, January 2020, we had the biggest bloom ever recorded. As you can see, how the bloom developed in December 2019, and a month later in January 2020. And during that period, there was a French cruise, which is part of this WASO project, the New Carbonate System Observations, which is a French initiative where they go on cruises and collect samples into this region. And this cruise went into that cruise and into that bloom and could look a bit more into details. But what they also found during that cruise was that when this bloom happens, it is a big region of oceanic carbon sink. Whereas when there's no bloom, it is in equilibrium with the ocean carbon source. And during this massive oceanic carbon sink, so this bloom draws down around 0.8 to 1 teragram of carbon in that month, which is quite massive. I want to start by revisiting the Bloom index. So what is a Bloom index? The Bloom index helps us to identify the amplitude and, of course, the timing of this bloom. So we define this by taking the chlorophyll A concentration into that bloom box, west box here, and we minus the chlorophyll A concentration into the east. So this is done because in this region, we also have a bloom in in winter. So by doing this this technique, we get rid of that winter bloom. And this is the time series that we are left with, which is what we call the bloom index. And as you can see, we have a bloom in 1999, 2000, a small one in 2002, a big one in 2008, and the one that I mentioned before, the biggest one in 2019, 2020. But what you can also see from this bloom is how variable the bloom is. For example, it occurred in 1999 and 2000, and in 2001, you don't see any bloom. Again, if you compare 2006, 2004 and 2005, there's a big difference. So, which is quite intriguing. It is not a seasonally recurring bloom, although it happens always in summer, in austral summer. Another quite interesting aspect of this bloom is how spatially variable it is. I showed the February 2006 before, but I'm also, if you look at February 1999, January 2008 and December 2019, you can see how different spatially the bloom is, and how sometimes it extends quite eastward, but sometimes it can be constrained closer to Madagascar region. And this, what we saw from the time series of the bloom index is also, you can also see it spatially here. For example, if you compare January from 2007 to January 2008, which is 1 year later, how different the spatial structure is. And again, in 2009, there's no bloom. So it shows you quite this this interesting seasonal and interannual, mostly interannual variability of this bloom. So the phytet algorithm, which is an algorithm based on fluorescence data from satellite, and this data was available from 1997 to 2010, and then discontinued. But from this time period, we could actually identify which dominant phytoplankton groups this Madagascar bloom is composed of, and we saw that the Prochlorococcus and Cynococcus are the most dominant phytoplankton blooms, and both are nitrogen fixers. So before going into the results, I want to just show briefly how complex the oceanic circulation is into this region. So this is so in this region we have the South Equatorial Current, which flows from Australia towards Madagascar, and then it bifurcates here to form the South-East Madagascar Current. The South-East Madagascar Current flows along the coast and then around the southern tip of Madagascar and flows towards the Agulhas Current as mesoscale eddies or as a flow. But this South-East Madagascar Current also induces a local upwelling south of Madagascar. In the other direction, we have the South Indian Ocean countercurrent, which flows from the west towards the east. But also, this South Indian Ocean countercurrent is quite shallow. And into the same region, we also have surface and subsurface intensified eddies, which flows from Australia towards Madagascar. So there's a lot of there's quite complex dynamical regime happening here with interaction of different current system and mesoscale eddies. Oops, sorry. Okay. So first we wanted to have a look at the mean vertical physical properties of the water column into the bloom region. So to look in which water column the bloom is being generated in. So if you have, so this figure here shows a mean austral summer sea surface salinity figure. And so what you can see is that there are very distinct water mass with colder, with fresher water mass close to the coast. And as you go away from the Madagascan coast, it gets more saline. So what we wanted to do is to collocate algal flows into this region with high chlorophyll A patches from satellite, which give us then how the mean vertical structure of the water column looks like when there's a bloom and when there's not a bloom. But we separate these two regions into a fresher and saltier side, so that we do not introduce any bias in the water mass structure. So in red, you could see all the algal floats which have been collocated with a high chlorophyll A patch, and in black you can see all the algal floats which have been collocated with no chlorophyll patch. So if we have a look of this mean profile, so this here shows temperature on the fresher side and temperature on the saltier side, and salinity on fresher and saltier side. So if we focus on the temperature on both sides, we can see that the bloom occurs into a shallow stratified mixed layer. The mixed layer is quite shallow, as we can see here. If we look now at the salinity profiles, in red is bloom, so we can see that the bloom occurs when the upper layer of the water column is fresher compared to when there's no bloom. So this is kind of an interesting aspect of this bloom, that it occurs into a layer of fresher water compared to when there is no bloom. So this begs the question, where is this fresher water coming from, and can this be connected then to how the bloom is being developed? And if we look now again at this climatology of sea surface salinity in austral summer into the region, we can see that the fresher waters are mostly aligned to the eastern coast and southern coast of Madagascar, which could potentially hint to the hypothesis that this fresher water is coming from the coast. So if I now focus onto this biggest bloom that I mentioned before, the December 2019, January 2020 bloom, and I repeat the same analysis, and we can again see that if we look at the salinity profile here, that the bloom occurs in a much fresher upper water layer compared to when there is no bloom. So we have now established that the bloom occurs within a shallow stratified mixed layer with fresher water on top, in the upper layers. And we also want to investigate if there is any possible links to other processes. And here I'm showing this sea surface chlorophyll A concentration, and this is Madagascar, the southern tip of Madagascar. So this upwelling cell here, like I mentioned before, is driven by the South-East Madagascar current, which flows here and then around the southern tip. So it's a current-driven upwelling, but it's also the wind forcing also contribute to this upwelling. And one of the hypotheses that have been put forward in a previous study stated that the bloom could occur because there is a high upwelling into this region, and then this high chlorophyll A patch at the surface is then advected towards the bloom region, which is more towards the east. So we test now this hypothesis, and so we develop a hypothesis testing model, and and from this, so here this is the coastal upwelling index based on sea surface temperature, and in red is when the coastal upwelling index can predict the bloom, and in blue is when the coastal upwelling index cannot predict the bloom. So what we found is that the bloom occurs when the coastal upwelling is at its minimum, which is kind of contradictory to what this previous study have shown. So the bloom does not co-occur with high upwelling into this region. So yeah, there is a decrease in current-driven upwelling during these major blooms of 1999, 2006, and 2008. So we also tested this hypothesis with the NINO 3.4 index. And here we found that the bloom occurrence actually coincide when there is a negative NINO 3.4, that is during La Nina phase. This is because usually the ENSO induces a wind stress curl anomalies over northwest of Australia, which could generate interannual variations into the South-East Madagascar current. So the main findings now is that from this observational aspect, at least, is that the bloom occurs within a shallow stratified mixed layer with lower salinity waters at the surface. And the blooms coincide with the mid-mature phase of La Nina years, and that also that bloom occurs during a reduced current-driven upwelling south of Madagascar. So putting all of this together, we come up with a new hypothesis, which says that during La Nina years, you have a stronger south equatorial current, which means that you have a stronger South-East Madagascar current. And this stronger South-East Madagascar current can retroflect without reaching that southern part of Madagascar. And it retroflects, it brings these coastal waters towards the bloom region. And by doing so, it could potentially advect nutrients, trace elements like dissolved iron, or even phytoplankton biomass from the coast towards the bloom region, contributing to the bloom. And if that Southeast Madagascar current does not reach the southern tip of Madagascar, this means that the upwelling intensity in this region is reduced, which is coherent to what we just found. So we put forward this hypothesis, and if we have a look now at sea level anomaly and geostrophic currents from altimetry, a composite during bloom and non-bloom, you could find an advection pathways from the eastern coast of Madagascar towards the bloom region, which does not seem to occur, which does not seem to be present during non-bloom regions. So we have this new hypothesis. And now we want to test it using a high-resolution coupled biophysical model. So this is the configuration of the model, which is based on CROCO and coupled with PISCES. And the parent grid is 1/4 of a degree, and the final resolution grid is 1/12 of a degree. And however, this is a version of the biogeochemical component does not include river runoff, because if you include river runoff, then you have an overestimation of nitrate and phytoplankton. And also there's no parameterization for nitrogen fixes. But we didn't want to look into the biogeochemical part too much, but we just wanted to look at the physical drivers using that bloom. So that's why these two aspects of this version of PISCES we used did not really affect our result here. So first of all, we want to look in the model if there is a phytoplankton bloom. So here I'm showing a mean surface chlorophyll A concentration. So this is averaged over the first 20 meters, and this is average between 20 and 40 meters. So what we can see in the surface, there is a hit of higher chlorophyll A concentration, but not that much. Whereas into the subsurface, you could potentially find structures which are quite comparable to what we see in observation. So, which means that we have a bloom, but it does not occur into the upper 20 meters into the model, but slightly deeper. And if we look now, if we repeat that chlorophyll A bloom index analysis with the model, we found that, so that is the chlorophyll A into the west box minus the east box from the model, we find, this is the time series that we have. And and from this, you could see these intermittent, these regions which are shaded with gray. You could see some higher chlorophyll A concentration during austral summer. And what we could also see is that they are quite erratic. So it happens into like 3 years here, and then doesn't happen for like a few years, which is quite similar to what we see in observation. Of course, the timing are not coinciding because this model is not assimilated, but we could see processes that could drive an erratic interannual variability of the bloom into the model. And now again, we repeat that vertical physical properties of water column into the model, and this gives a similar result of the bloom occurring into shallow stratified mixed layer with fresher waters into the upper layers of the bloom. So these are quite similar vertical structures which are associated with the bloom in both observations and in the model. And if we look now at the sea level anomaly with with overlaid geostrophic currents averaged over the bloom years that we found into the model and non-bloom years, we could see that in the model during bloom events, this region seems quite more energetic compared to when there is no bloom. So which could potentially also create pathways for eastern Madagascar coastal waters to move into this bloom region here. And the cool thing about the model, of course, is that you could do fluxes of nutrients, for example. So we did this with nitrate. So this is our bloom box into the model, and we tried to quantify the nitrate fluxes coming from the coast compared to the fluxes coming from underneath the bloom box, that is upwelling fluxes. So here I'm showing results for 3 different blooms, but let's focus on 2005. So here in red, you could see how the chlorophyll A concentration increases during austral summer, which is representative of the bloom, and at the same time, the nitrate concentration increases. So this nitrate concentration, we can decompose it and see if it's coming from the coast or from the vertical. And if in blue is the nitrate fluxes from the coast, and in red is the nitrate fluxes from underneath, that is upwelling. So what we can see is that in 2005 we have enhanced nitrate fluxes coming from the coast, but we also have some nitrate fluxes coming from underneath. So it could be a combination of both, creating initiating this bloom. If we look at 2008 now, you could see that the nitrate fluxes is predominantly coming from the coast. So which means that there is a potential advection of high nutrient water from the East Madagascan coast into the bloom region. And if we look at 2010, first of all, the nitrate is mostly coming from the coast, and then the bloom is sustained by nitrate coming from from underneath, which could potentially hint at a possible mesoscale eddy into this region, which is uplifting nitrate from the deeper waters. And all of these nitrate, every time there's nitrate fluxes coming from the eastern coast, we could see that there is a signature of fresher waters into this region. So this is all aligning now, which we found is that the input of high nitrate concentration is predominantly from the coast, and these are associated with a low salinity water signature. And in the model, we found that 6 out of the 8 simulated blooms occurred during a time period of enhanced nitrate fluxes from the coast, which is aligned to our observations. Another interesting aspect of the model is that we could actually reproduce a bloom which was triggered by a cyclone activity into this region. So this is the bloom into the model in 2004, and you can see that so the bloom, the chlorophyll A concentration increases, also the nitrate increases, and the nitrate seems to be mostly coming from the vertical, from the deeper layers. So which is because when there is a passage of a cyclone over this region, this could create a negative wind stress curve, which bring a deeper, colder, deep, nutrient-rich waters into the surface. And if we look, this is from this is a wind stress curve from observations, and this is a wind stress curve from the model. And we could see that this negative wind stress curve is reproduced into the model, and this is linked then to this higher chlorophyll A into this region. So the model actually correctly reproduces a bloom which is triggered by atmospheric faulting. So this brings an additional piece to the puzzle. So from an observational aspect, we stated that an early detachment of the South-East Madagascar current before it reaches the southern tip of Madagascar can advect nutrients, trace elements, or phytoplankton biomass from the coast into the bloom region. And we've now tested this new hypothesis with a model, which also suggests that the simulated South-East Madagascar bloom occurs concurrently with an early detachment or retroflection of the South-East Madagascar current. Just a few more slides before I finish. So this is a more recent work, well, not so recent anymore, which was done after my work, and which shows that the East Madagascar current actually have three forms have three end forms. For example, the early it has an So it has an early retroflection when it retroflects without reaching the southern tip. There's another form, retroflection, which is called canonical retroflection. It grounds the southern tip of Madagascar and then retroflects. And there is another form when there is no retroflection at all. So this study found these three forms of the East Madagascar current, and they also found that during when there is no retroflection, for example, when there is a retroflection, there is a reduced upwelling south of Madagascar, which is again aligned to what we found. And so what we did was we also took all the events when there is no retroflection and then did a composite of the surface chlorophyll A concentration during that event. And what we found is that so whenever there is a retroflection, we have a high chlorophyll A patches into this region, which is then called the South-East Madagascar Bloom. So this composite of surface currents and chlorophyll A concentration during austral summer of events of early EMC shown that it could be a big contributor to this austral summer bloom. And yeah, I think, yeah, I will stop here now. Thank you very much for your attention.
Awesome. Thanks so much, Femi. That was a really interesting talk. And we have one question in the chat so far. And if anyone else has any questions, please feel free either to raise a hand or you can post it in the chat. But so Femi, the first question from Kumar Das is how can one find bloom events in coastal areas? So I think that's talking to the detection of or how you classify blooms.
Yeah, so let me go back to one of the slides maybe, and or maybe this one. No, this is too. Okay, this one. But so coastal areas, I would presume that the question is aligned to these coastal areas, or yeah.
Yep.
So how can we find bloom event in coastal areas? But coastal areas are usually high chlorophyll A regions in any case, because you have a lot of river runoff or river runoff towards the coast, and this brings nutrient-rich waters along the coast. And if you focus, maybe if you look at this map and on the coast of Madagascar, we can see all around the coast there is this red. So the water is shallower, so there's more direct sunlight, more nutrients, so there will always be a lot of chlorophyll A, high chlorophyll A concentration happening. And then of course, since the chlorophyll A concentration is always higher in this region, that you could potentially find a mean chlorophyll A concentration, and there would be events. For example, if there's enhanced rainfall into this region, there will be even more river runoff. Then your chlorophyll A concentration will be even higher than your mean or median, which could potentially be identified as a bloom. But in general, I would say coastal areas are bloom regions. Here, it's a bit different. It's more open ocean where it's quite oligotrophic. And of course, there's sunlight, but there's not much nutrients, and there's not always nutrients. So this brings to the interesting aspect of open ocean blooms.
Thanks, Femi. Yeah, I mean, Denisha has a hand up. I just wanted to follow on from the question because it relates. So, the term bloom, I guess, is that dependent? It's region dependent on. It's kind of you've set your own threshold on a bloom, and a bloom here potentially is not considered a bloom in a more chlorophyll A rich region. Is that correct?
Yes, no, I totally correct. For example, if we look at this maybe bigger picture here, if you look at the Benguela region, right? The Benguela region is almost always, I think, productive region. But so it's do you actually call it a bloom, or is it because if you call it a bloom, then it's a bloom which happens all year long, right? So I think it's just part of the seasonal and interannual cycle. So I will not maybe call it a bloom, but I will just call it a very productive region. In general, eastern boundary upwelling regions are, to my knowledge, at least, not called blooms. Again, here for the Canary Mauritanian upwelling region. I'm not too familiar with this region. I guess there's people in the audience which are more familiar with this. If we talk about Southern Ocean, you always have this high bloom, high chlorophyll A pact, which migrates south and north along the Antarctic circumpolar. No, this is the Agulhas return current. Sorry. So this is the Agulhas return current, which is always, I think, always high. So do you quantify it as a bloom? I'm not sure. But if I focus here, or sometimes you have a bloom into the, if you look into the Seychelles thermocline ridge, which is also sometimes where you have high chlorophyll A, even higher chlorophyll A into the surface, you could potentially call it a bloom. And also in this region, because it's usually oligotrophic, there's nothing happening, and then just once in a while you can just find like an explosion of a lot of phytoplankton cells. So yeah, it's a good question, and I think it doesn't have a really clear answer.
Yeah. So Denisha has her hand up, but there's a follow-up question. It should be a quicker one. It's how to download the data. So I guess what data did you use then?
Yeah, so this data here is from the European Space Agency, the ESA Ocean Color OLCCI, something like this. So these are satellite data. And so this product actually, so you can have daily, 8 daily or monthly data. Of course, the daily data have more cloud covers. But this product so if you go on the ESA Ocean Color, you will easily find it. And what it does, it combines chlorophyll A data from the MODIS, the MERIS, and which one? The CWIFS satellite. So to create kind of a combination of to get a higher to get more data actually. So yeah, that's the ESA Ocean Color. Yeah. Should have mentioned that. Sorry.
And Denisha, you have your hand up.
Yeah, thanks, Semi. So there's quite a strong link with salinity, but I always wondered if there's a link between chlorophyll and temperature. Like, for example, during La Nina, this region is usually much cooler. Does this play a direct role? I'm not sure.
So if there's a link with temperature and the bloom, right? I'm not sure. So you said, Danija, that during El Nino, this region is much cooler?
Yeah. So
Oh,
thinking of
okay.
salinity, lower salinity, would temperature maybe might play a role in the generation of the bloom. I'm not sure. Yeah, it's not always a direct link, I think, between chlorophyll and temperature.
Yeah, so at least from these ones, I could not. I mean, from the temperature, I could just. So it's kind of opposite here. So I would. Yeah, it's really hard to say if it's colder or not, or less colder during bloom and non-bloom, and but yeah, I can't really say. But I remember. I know that the bloom are actually confined. So if you look at this filamentous structure of the bloom, they are quite the bloom. The extension, the latitudinal extension, are quite confined to sea surface temperature gradients, at least. But what does that tell us? I'm not. I'm not sure how to link that to actually sea surface temperature. It could just be that.
Just a lot of mixing.
Yeah, it could be the mixing. Could be I don't know, some vertical velocity happening at these gradient of sea surface temperature, or they are just restricting it spatially. Yeah, I'm not sure. Yeah.
Okay, thank you for me.
Okay. Femi, we have another question in the chat from Baskara, and it asks how to differentiate bloom chlorophyll A concentration ranges from coastal waters to open ocean waters. So I guess that's quite similar to what we've already answered. I don't know if you want to add anything to that.
Bloom range from. Yeah, I mean, I think you you don't really try to differentiate it. It's two different regions with two different forcing mechanisms, and like I mentioned before, like a mean concentration into a coastal region will probably always be high compared to a mean concentration of chlorophyll A into an open ocean. So yeah, it's all about the mean environment, I would say. The mean environment is what dictates bloom and non-bloom, and yeah.
Cool. And there's another chat question for you, Femi, is why is the bloom form year by year increase? So I guess looking at why, is there any reason why the 2021 was so much bigger?
Yeah, I mean, that would be cool to know why. I don't know. There's no yeah, we did not, at least from my work, we did not really manage to put a finger on exactly what's creating the amplitude, what's driving the amplitude of this bloom. So, and like I mean, I also want to say that we did not solve the problem of the bloom. Huh? There's a few hypotheses. There's a lot of hypotheses that have been put forward, and we tried to revisit this hypothesis and also to bring maybe another piece to the puzzle. But for example, yeah, it's hard. It's really hard to really find. I personally don't think it's one single mechanism. I think it's a couple of mechanisms which have to align at the same time for the bloom to occur, but also that this couple of mechanisms could potentially also be responsible for that for the difference into the bloom amplitude. And yeah, I mean, like the 2019-20 bloom is really, really massive compared to the other ones, and it would be good to see why this is the case. Yeah, I don't have the answer for this.
Yeah. And I have a question as well, but just following up on what you said, it's quite interesting. If you look at that previous slide of the bloom events, it actually looked like the blooms were decreasing in time up until that 2020. So there was that big gap from about 2009 to 2020 where there was basically no bloom, and then one giant one. So it seems, like you say, very, very erratic.
Yeah.
But sorry, my question, Femi, is, so for that 2021 in particular, there looks like there's a big connection with the Gullis current. The bloom, the high chlorophyll concentration seems to stretch from the Gullis current all the way across in January. And so it's quite surprising to me, because you talk about the early retroflection, but it seems like that nutrient-rich water is maybe also getting advected into the Gullis current. Is there any comment on that?
Yeah, so yeah, but first I think if we look at the 2000 and yeah, this is, so the bloom started a bit early, December 2019, and then it really peaks in January. So in 2019, if you look at it, you could see this really filamentous structure, but there's I don't know, there could be there's not a real connection with the Agulhas current here, right? But it's more here, and then there are some mesoscale activity which could also induce this high chlorophyll A patch. But if you look at this picture here, but this is now this is a I would not I don't want to say it's connected to the Agulhas current, or I don't want to say it is connected to the Agulhas current, because I think here my color bar, it might be a bit not stretched enough. So maybe if I increased my color bar, then you could see that there is kind of a disconnection. So I cannot really say if it is connected or not connected, because maybe I saturated my color bar a bit too much to show the how big the bloom is.
Yes.
But yeah, but if it's but again, if you look at this picture here, you could see there's this big patch here, and then there are more like a mesoscale, like tiny mesoscale beaches here. I don't know if you can see it properly, but there are like these rings here with low chlorophyll A into the lower chlorophyll A into the center. Yeah, there could be a connection, or there could not be a connection. So yeah, and if there is a connection, let's assume there's a connection. Maybe the bloom starts here, and then there's an advection of phytoplankton biomass all the way to the Agulhas current. But yeah, it's a long way, so it's hard really to imagine. Yeah.
Yeah, Denisha had a comment on that, as on the 2021 as well. She says that it looks more widespread, maybe more atmospheric, stronger winds would be interesting to check.
Yeah. Sorry.
Oh no, that continue then.
Yeah, exactly. I think it would be very interesting to see. I mean, it's yeah, could be winds, could be, like I say, could be combination of different mechanisms. What else? Yeah, I mean, it's quite massive again. There must be a signal. So when I did this work, I must maybe say this. When I did this work, I think I stopped the time series stopped in 2018 or 2016, I think. So, and then I updated this time series. So most of the analysis that I've shown do not include this big peak here.
Right.
The only analysis that include this big peak is when I look at the mean vertical structure during this look at the at the Argo floods during that time. But all my analyses do not include this 2020. But it is that big that I think there should be a connection. If someone is willing to look into it, there should be a connection somewhere. I would love to do it, but yeah, because of timing, it's a bit.
Yeah.
Yeah, it could be atmospheric, could be a combination, or yeah, atmospheric. I don't know.
Okay. And sorry, Femi, just last one. Did you look at rainfall for increased runoff?
Yeah, I did look at rainfall, but if I remember correctly, there was no real connection to the increased runoff. To the bloom, at least. I mean, if you look at the time series of rainfall, I would, but I did it mostly over Madagascar region and try to link at maybe there's more rainfall into into December, then the following January there's a bigger bloom. There's a bloom happening. If there's not enough rainfall, there's not that big of a bloom. But it's good that you mentioned it, Matthew, because I think for some reason I think rainfall is a very important factor here. But and not only rainfall on land, but also rainfall onto the ocean, because rainfall on the ocean can create quite a stable haline stratification. So lower surface salinity, and then this haline stratification, which could be for like, I don't know, maybe could be 5 meters or so in depth. This haline stratification could also play a role in this bloom, because some phytoplankton groups or organisms, I'm not really a biological person, but I think some phytoplankton groups favors these haline stratification to really grow. So yeah, I think rainfall is important, but not only on land, but also on the ocean. But unfortunately, I did not look into it.
Yeah. There's only so much time that everyone
Yeah.
has. So we have another question from Tallulah. She asked, could there be a relationship between the positive IOD and the blooms?
Yeah. So when I did this hypothesis testing model here, so to test these hypotheses, I think so. I did this for IOD also, but it was not super clear. For example, so here we have the time series of the Nino 3.4 index, and in red is actually when the negative Nino 3.4 coincides with the bloom, and in blue is when the positive Nino 3.4 coincides with the no bloom. So all the dots actually, no, forget about the crosses, all the dots actually shows that the time series could potentially explain the bloom, the bloom and non-bloom events. And I did similar thing for the IOD, Indian Ocean Dipole, positive and negative. And if I remember correctly, there was no clear outcome. There was no clear connection. But this also brings to the question that there seems to be a connection between El Nino or Nino 3.4 and IOD. So yeah, it could also play a role, but yeah, it's not that clear. Yeah.
Cool. Thanks, Femi. And then so there's one more question from Kumadas, and I thank you. I see that you shared your email address, Femi.
Yes, I did.
So one bloom form, how much a day is staying on the coastal surface? Sorry, to me, the question's not very clear, but do you have any, can you see the chat thing?
Yeah, I can, but I also do not really understand the question. Sorry. Can you maybe rephrase it, please?
You're welcome to unmute as well and ask your question if you'd like. Kumar Das. Not sure if he or she can hear us, or is the life thanks Himashu. So Femi, do you want us to talk about the lifespan?
So yeah, the lifespan. So it's again, this is something that is very variable. I can't remember. I think it was a few weeks, but it's not a specific lifespan. I think, for example, here let's oops. If we have a look at this Bloom index here, let's compare 1999. So all the gray shaded areas are where there was a bloom identified from this Bloom index here. So here, if you look at 1999, okay, it peaks in, I don't know, March, January, March, and then goes down really fast. But if we look at 2012, it peaks quite rapidly, but takes a bit more time to dissolve. I think the lifespan was around 6 weeks upward, 6 weeks to, I would say, 10 weeks, but there's no, like the spatial variability is very erratic, like the interannual variability is very erratic, the lifespan is also very erratic. So it's yeah, there's no fixed answer to a fixed lifespan. It varies by a few weeks.
Cool. I think that's a good answer. I think there's no more questions in the chat. Does anyone have any last questions for Femi? If not, yeah, thank you very much, Femi. That was really, really interesting, and yeah, great to hear about the work.
Thanks, Matthew.
Cool. Thanks all, and thank you for attending, everyone.